We are continuing with the lecture notes from chapter 20, beginning with slide number 16. We're going to talk about the flu virus. Flu viruses have glycoprotein spikes called H spikes and N spikes. H stands for hemagglutinin and N stands for neuraminidase. The H spike allows the virus to bind to the receptor on the host cell, and the N spike attacks the host cell's plasma membrane, so that the plasma membrane will engulf the flu virus and begin making new flu viruses. There are 16 different types of H spikes and 9 different types of N spikes, which causes the different varieties of the flu. And the vaccines are made, flu vaccines are designed for the H and N spikes that are particularly found within that particular flu season. All right, so what we're looking at here in picture A is the original human influenza virus, and you can see the N spikes have kind of a round surface and the H spikes have a pointy surface so they can attack the plasma membrane of the host cell, which is the cells of our respiratory tract. And then after mutation that occurs in the virus, in this particular instance, the H spike has been modified. And so that happens each flu season, is there are mutations in the flu virus, and the new vaccines have to be made to adjust for those mutations. If you look at picture B, it just shows you how if the respiratory fluids of birds and humans come in close contact, how the viruses can actually mix, and you can end up with a combination of spikes from the human and the bird flu that will allow humans to be infected with a bird flu. And that's how it happens. A lot of times, the only way you're going to get that close with humans and birds is when, like, people working in chicken houses or turkey houses, coming in contact with the respiratory secretions from the chickens and the turkeys. Now, emerging viruses are new or previously uncommon illnesses. Examples are the coronavirus, we refer to as COVID, Zika, the Middle East Respiratory Syndrome or MERS, AIDS, West Nile encephalitis, Ebola hemorrhagic fever, and the bird flu or avian influenza. These viruses can emerge for several reasons. One would be that the virus can extend its range, and the other can be genetic mutations, such as what we see with the flu. Viroids and prions are other substances basically that can cause disease. They are not viruses, and they're also not cellular. So they're a viroid. Oh, look at the very top. Viruses are best known for causing infectious diseases in plants and animals. Viroids are naked strands of RNA that are responsible for many crop diseases. And prions are protein molecules that have misfolded. They have a tertiary structure. Remember, that's the three-dimensional structure of the protein. But that tertiary structure has misfolded. It is faulty and contagious. Prions cause TSEs, which are neurodegenerative diseases that destroy nerve tissue in the brain. They are untreatable and fatal. Mad cow disease is one of these. It is a BSE, which is bovine spongiform encephalopathy. In section 2, section 20.2, we'll talk about prokaryotes. Prokaryote, we have two basic types of cells in biology. There are two types of cells. One type of cell does not have a nucleus, and it's called a prokaryotic cell. The other type does have a nucleus, and it's called a eukaryotic cell. Again, we learned this in Bio 111. There are also 3 domains of living things. 2 of them are bacteria and archaea, and the third is eukarya. The domains bacteria and archaea are composed of organisms that are prokaryotes. That means that they are unicellular. They're only composed of one cell, and their cells do not have a nucleus. Louis Pasteur, who was also the first to use the word virus, showed that a previously sterilized broth cannot become cloudy with growth unless it is exposed directly to the air. This is an illustration of his experiments when he used a flask that had a straight neck, as you see up here at the top, and he boiled the contents of the flask to sterilize what was inside. Then he took the stopper off the flask and exposed the flask to the outside air. Then he restoppered it. 89% of his flask became cloudy and showed bacterial growth. He did the same experiment inside the building, and it reduced the growth. It was only 32% that showed growth, but there were still bacteria that were getting inside the flask, even inside the building. So he invented this curvy neck for the top of his flask, and he didn't even use a stopper. He just he just had a flask with this S-shaped neck. He boiled it, and he saw 100% had no growth with these special necks that he invented. So what happens is air, there's no picture. Okay, so air still enters this open neck, but the bacteria from the air collect here in this depression, and they don't travel up the neck. So again, the bacteria are settling in this depression, and they aren't going to travel up the neck. So that is the reason that pasteurization works to sterilize a liquid such as milk. Okay, so that's how pasteurization works, basically. Now, as far as the structure of prokaryotes, which would be the members of the domains archaea and bacteria, they do not have a membrane-bounded nucleus. Their DNA is found in the nucleoid region of the cell. They have a cell wall outside the plasma membrane. Some have flagella and can move. All of them lack membranous organelles such as mitochondria, chloroplast, lysosomes, endoplasmic reticulum. Those organelles are only found in eukaryotes. And they may have accessory or extra rings of DNA called plasmids that have extra genes located in them. So this is an outline of the structure of a prokaryotic cell. Prokaryotic cell will have a cell envelope composed of the plasma membrane on the inside, surrounded by a cell wall and a glycocalyx, which can be like a slime layer, or it can be tougher. It can be a capsule. And then the cytoplasm on the inside will contain the nucleoid region where the DNA is found, ribosomes, and thylakoids only in the cyanobacteria, which are photosynthetic. Then we have some structures containing appendages such as flagella for movement, conjugation pilus for exchanging DNA, fimbriae, which will help the bacterium attach to other bacteria and to the substrate that it's growing on. Here you see the features of prokaryotic cells. You can see the fimbriae, which help with attachment, the nucleoid region containing the DNA. The DNA is found in a single circular chromosome. It's just really long, so it's all folded up in here. The conjugation pilus allows for the bacterium to inject some of its DNA in the form of plasmids into other bacteria. And then the flagellum, this one has, it looks like 3 for movement. And you can see the layers of its surface. The plasma membrane would be on the inside, then you would have the cell wall, and outside that sometimes there will be a capsule. Reproduction in prokaryotes is entirely asexual through binary fission, which means to split in half. The notes say that the generation time is as short as 12 minutes. That means it can happen as quickly as 12 minutes for one bacterium to reproduce and produce 2. And this binary fission is basically a form of cloning. Prokaryotes are haploid because they only have 1 chromosome, and diploid is, for example, human cells are diploid, and they contain 2 copies of each chromosome, but prokaryotes are all haploid because they only have 1 chromosome. Some bacteria can form endospores, such as the botulism, the Clostridium botulinum bacterium that causes botulism, can form endospores which are resistant to unfavorable conditions, and then they will become active when conditions become favorable. This is just an image of binary fission taken with an electron microscope. There are ways that bacteria can undergo genetic recombination and to where they can have variety, genetic variety. One process is conjugation, which is when the one bacterium injects DNA into a recipient cell, and it uses the conjugation pilus to inject the DNA into a recipient cell. There's also transformation, and that is a process where the bacterial cell can pick up DNA from its environment, often from dead bacterium in its environment. Then there's transduction, which is where bacteriophages, which are viruses that only infect bacteria, can actually carry DNA from one bacterium to another. The virus serves as a vector. The bacteriophage virus serves as a vector. Now we're going to move on and talk about bacteria in section 20.3. Bacteria are the more common type of prokaryote. The archaea are not as common. Bacterial cells are protected by a cell wall that contains the chemical peptidoglycan. The peptidoglycan is the chemical that absorbs the stain when we stain bacteria in the Gram stain procedure. There's a lot of ways to stain bacteria so you can see it under a microscope, but the first type of staining, the most common type of staining that we use is the Gram stain. So Gram-positive bacteria will retain a dark purple stain, and they will appear dark purple under the microscope when we look at them under the microscope. They have a thicker peptidoglycan layer that allows them to retain that stain. Then Gram-negative bacteria won't retain the stain because their peptidoglycan layer is thin, not thick. We have to stain them with a different stain, and they will appear red. And actually, that stain, the stain is red, but they actually appear pink under the microscope. Gram-negative bacteria have a second plasma membrane that blocks antibiotic drugs. So oftentimes Gram-negative bacteria are more difficult to treat. This is a picture of gram staining and what it would look like. In gram staining, you start out by applying a dark purple stain called crystal violet to your bacteria on your microscope slide. And then you add iodine, which helps to lock in the stain. So all of those bacteria that are gram positive will retain that purple stain. But if they're gram negative, after you do this, third picture shows a process called decolorizing. When you decolorize by adding alcohol to the slide, the gram negative bacteria will lose the stain and the gram positive bacteria will keep it. So you have to add a red stain called safranine, and that red stain will be absorbed by the gram negative bacteria. And what this slide will look like under the microscope is the gram positive bacteria will appear purple and the gram negative will appear pink. And we'll move on. This is where we'll start in the next recording.